So yes, I'm Yoselin Benitez-Alfonso. I am the Associate Professor at the Plant Science in the University of Leeds, and I have been coming here to the Gatsby Plant Science School for 10 years, about 10 years, as a tutor. So this is a great honor for me. It's like a promotion. I got promoted to be a plenary lecturer. And I want to start with this view. This was from the Caminito del Rey, and where I was last week in the cell wall meeting. And it's showing basically a stream of water running between walls. Not really similar, but Plasmodesmata have some similarities to a stream of water running between walls. So they are basically cytoplasmic connections, so that mediate the communication of molecules between neighboring cells. So here you see in a cell, in a plant cell, you see these locations, in this case, are marked by a Plasmodesmata protein, which are in a specific microdomains of the cell wall. And if you look in an electron micrograph, this is what you observe, just basically a tomography. You can see a membranous channel that connect two cells and traverse the cell wall, just like this river here in the Caminito del Rey. So why plants need Plasmodesmata? Probably I don't have to convince anyone here, but plants are under the pressure of many stresses while they are growing, since they are germinating until their adult time. They are under the stress of having to cope with differences in temperature, in light conditions, with pathogens such as insects, with the lack of nutrients, with the lack of water, and even with ourselves and with a dog pooing in their roots. So all of them make plants needs to be really fast to adapt to the environmental conditions, to what is thrown at them. So they need to transmit those signals very fast and then respond to them very fast, or they are dead. So in plant cells, we have a surrounded of a rigid cell wall. And that's what most of you know about the main differences between plants and animal cells. So these kind of wall barriers for this communication. And that's the reason why plants need these channels, the Plasmodesmata, which allows the cell wall to don't exist in certain spaces and allow for the cells to communicate macromolecules and such as proteins, RNAs, metabolites, hormones, and other macromolecules. So the video that I'm going to show now is showing a plant under the UV light, and a virus protein, which is infected in one of the source leaf of this plant. And I want you to look at it and see how the virus protein moves from the source cell into the zinc tissue, into the new leaf. And this virus protein is fused with GFP. Can anyone tell me which color the plant will be under the UV light? Yeah? No? On the UV light, that plant will be red because of the chlorophyll, and the virus will be green because of the green fluorescent protein. So here you see the tobacco plant growing. And here in this source leaf, you see this yellow light. That's the green fluorescent protein that has been infected here. And you see it moved very fast into the new tissue. So this is basically possible because of Plasmodesmata. So virus has been tricky enough and smart enough to exploit these channels to allow their spreading between neighboring cells and into the phloem and into the zinc tissue. So clearly, Plasmodesmata are important, and this is what my group is studying. Basically, we carry out cross-disciplinary research to dissect what are the structural properties of these channels and how they allow for communication, but also understanding how we can exploit this knowledge into creating new solutions for crop improvement or biosustainable materials. And these are my team, the people that really do the amazing work that I'm going to be talking today. And we are funded by several organizations, more recently UKRI. And please contact us if after this talk you have any questions, but also check our website to meet all these amazing people that I am mentioning here. So this project that I have with UKRI Future Leaders Fellowship really focus on Plasmodesmata walls and is in the topic of mechanobiology. So how these walls are affecting the structure of the channel, the transport through the channel. And we do this by dissecting the properties of the wall by using different techniques and from different fields, such as structural cell biology, polymer physics, genetics, and computer simulations. My team is actually dispersed within all these areas of research. So some members of my team are working specifically on the cell-to-cell communication topic. Others are looking at how these cell walls can actually give us clues to bio-inspired materials. And most of them, actually, are working on how this cell-to-cell communication impact the plant responses to environmental signaling, organ growth, water and nutrient uptake, and symbiosis. So today, I want to give you just a glimpse of the type of research in these three different areas that I am mentioning. So how we gain knowledge on PD wall structures, their properties, and their regulation. How we use this knowledge to identify strategies to target Plasmodesmata for crop biotechnology. And finally, I will give you some insights in how the knowledge that we acquire from these cell walls components can help us in design materials. So we're going to be dividing this talk in questions. And the first question that I want to answer in this first part of the talk is how the structural and mechanical properties of cell walls regulate transport through that channel. Cell walls are really a complex mixture of polysaccharides. So they are made of pectins, cellulose, hemicelluloses, soluble proteins, and all these polysaccharides makes the cell wall very complex to study. So how we access information on what are the specific characteristic or features of cell wall Plasmodesmata. So we have decided to opt to one of our molecular props, and these are antibodies. So antibodies in our lab are generated in rats, and by injecting an antigen that is one of these cell wall components. And they are very specific in detecting this cell wall component in cell walls. So the second major player in this cell wall characterization is to have a plant or a cell wall structure that is big enough so that we can observe Plasmodesmata, and this is tomato pericarps. So we isolate cell walls from these tomato pericarps, and here you can see how these cells are really pushed open, full of water, but the Plasmodesmata aggregates into picked fields that are at least somewhat observable under the microscope. So these two players allow us to use these molecular props to understand what are the properties of the cell walls around picked fields and outside of these picked fields. And picked fields are only complex Plasmodesmata. So if we look in immunolocalization, which is where we use these props, we can see in green here that these picked fields are rich in certain types of pectins. But more importantly, because you will see that this is very constant throughout my talk, they are enriched in a cell wall polysaccharide that is named callose. And callose accumulates inside of these pit fields and around the channels. So notice that this is the cell wall laying on the top, but really in the width you should be able to see something like this. And callose is also accumulating in a different patterns depending of the type of pit fields and depending of the antibody that detect this polysaccharide. So this is not only real or seen in tomato pit fields. Also, callose accumulates at cell wall microdomains in miscanthus and in Arabidopsis leaf and in tobacco roots. So it's very widely known that callose is actually associated with the Plasmodesmata channel. And if you don't believe this immunolocalization, because you cannot see the Plasmodesmata, you can actually do electromicrograph with immunogol staining, and here you can see a cell wall and a Plasmodesmata. And maybe you cannot see it from there, but there is some dots there, and that is the callose that is accumulating around this channel, which in this case is marked by immunogol particles. But what is callose? Well, callose is integrated in cell walls, and here you can see in pontamin red the cellulose microfibers. If I induce callose, you can see that this is stained in blue with a dye that is named aniline blue. And it's really intrinsically complex with the cellulose microfibers. Callose is synthesized by a cell wall enzyme named callose synthase that is very similar again to the cellulose synthase and is accumulated in response to pathogens, to fungal pathogens and bacterial pathogens, and is disrupted or induced if I disrupt the cellulose with chemical such as isocyanates. So another way that we have to actually look at what is around Plasmodesmata and what cell wall components are around Plasmodesmata is by looking at the proteome. So by isolating which proteins are actually associated with the membranes of these channels. And for that, we and others have carried out proteomic screenings, isolation of the membranes, and this is a little bit of the protocol that is used. Isolation of the membrane that is associated with the cell wall, and using mass spec identification of the peptides and reconstruction of the proteome. So we recently used several of these proteomes and investigated from the signals or domains that are conserved in the different proteomes that has been identified. This has been done in Arabidopsis, but also in poplar and in tobacco. And if you want to know more about the proteome, check GitHub, where we have this flow already implemented in a kind of an in silico proteome that is named PIP1. So what allows us to do the proteome is to identify which enzymes or which proteins are associated with Plasmodesmata that might have some kind of cell wall activity. And hopefully, we will identify callose degrading and synthetic enzymes because we know that callose is at Plasmodesmata. And that's indeed the case. So using the proteome, we could identify callose synthase, PD or Plasmodesmata beta-1,3 glucanases, which has the callose degrading enzyme, but also other proteins that contribute to callose regulation. So in fact, we can identify a number of proteins, so many in the proteome, that we need to do an interactome between the genes expression of the encoding these proteins. And again, we could identify callose synthetic, degrading, and regulatory enzymes, and these are co-expressed with enzymes that modify pectin, cellulose, and other polysaccharides in the cell wall. So to verify that indeed the enzymes are at the Plasmodesmata location, we use immunolocalization, again, in this case, together with a fusion protein in which these proteins are fused to a fluorescent probe, so we can see where they are located. And here you can see a callose degrading enzyme, again located in a very pattern around the cell. Here you see a cellulose modifying enzyme and expansing and other xyloglucan modifying enzymes located at Plasmodesmata. Specifically for callose, we can see in Arabidopsis thaliana the localization of this PD-located beta-1,3 glucanase, again, very, very, very tightly combined with the Plasmodesmata location. So now we have identified enzymes that modify callose. We have identified that callose is at Plasmodesmata. So what is the function of callose at Plasmodesmata? So for that, what we use is a genetic screen and identify how symplastic reporters, so reporters that tell us the movement of molecules around Plasmodesmata is affected when I modify callose. And this is seen here, mutants in PD-located beta-1,3 glucanases, such as PDBG1,2, are restricted in the transport of GFP. In this case, GFP is driven by a companion cell, a specific promoter, so a promoter that is expressed internally in the vascular tissue, and GFP can move through Plasmodesmata from these companion cells into the neighboring tissues, including the cortex. So in these mutants, you can see that there is a restriction in the expression of this GFP that now is just constricted or restricted to the vascular tissue. So not surprisingly, modifying symplastic communication through modifying callose also lead to really cool phenotypes. And this is seen here in lateral roots. So the mutants form lateral roots in bundles or kind of in twins, which suggests that perhaps there is a signal that is required to move in these mutants to determine where is the patterning of lateral roots, in this case in Arabidopsis, but also in other plants. So callose regulation is really important for Plasmodesmata communication. And the hypothesis so far coming up from this research is that callose accumulates around Plasmodesmata and restrict the transport of molecules through. That's the case because when we modify callose, when we increase callose by mutating these beta-glucanases, then we get a restriction in transport. But what happens when we ectopically express these callose synthesis? Well, what happened is the same. So in this case, we evaluate this, or I'm showing a picture in which we evaluate this by expressing GFP in the outside cell layer of the root. And in plants control, GFP can move from this epidermal tissue into the internal tissue, but this movement is restricted when I ectopically express this callose synthase 3 line, which is an activated version of the callose synthesis. Again, this must lead to some phenotype, right? And indeed, this is the case. So when you induce, in fact, you cannot express the activated version constitutively, because if not, the plant dies. But you can do it in an inducible system. So we do it in a way in which estradiol is used to induce this enzyme. You get an overaccumulation of callose, but also a restriction in plant growth in comparison to wild type, suggesting that indeed callose is a key player in regulated transport through these channels. But what are the properties of callose? How callose do that? And just spoiler alert, I don't know. So what I'm telling you here is ongoing research. And we have investigated this, again, using tomato fruits because it allows us to gain these big cells. And what we know is that during tomato fruit development, it seems to be peaks of callose deposition. And we investigate how basically these peaks of callose deposition coincide with the mechanical properties of the cell walls using FM nanoindentation. And for some of you that might not be related with this technique, we basically use something to poke the cell wall and see how they react. And from there, we get what are the properties of those cell walls, so how mechanically they behave. With FM, you can also get beautiful pictures of the cellulose microfibers in the different stages of tomato development. As expected, the cellulose microfiber seems to be thinner at the end or later in the development, and this might be related with the part in which tomatoes start to soften it up. And we can just focus more closely in those cell walls and look at the pit fields and look how cellulose is organized around these pit fields and then extract mechanical behavior around these pit fields and outside of these pit fields. And from this data, we can infer that callose kind of soften the cell walls around Plasmodesmata, contrary to what we expected, to be fair. So this image is starting to emerge. So basically, callose is a key player surrounding the channel, controlling the size of the cytoplasmic sleeve, and this size of the cytoplasmic sleeve, which allow basically for molecules to move from one cell to the other. But this is happening together with a plethora of processes that occur in the cell wall, including the cellulose synthesis, but also xyloglucan modifications, mannans, pectins, and all these modifications might interact with the cell walls around Plasmodesmata and really modifying also the properties of callose around these cell walls. And this is a field of research that is, again, focus of my project, which I am in my second year of this project starting. But let's move on, because I want to give you another different flavor of the research that is ongoing in the lab. Specifically, I want to tell you how we use this knowledge on Plasmodesmata cell walls to find strategies to sustainably improve food security and to deal with climate change. And this cannot be done alone. Climate change really poses impossible challenges to one research group. So we do it in a network, and this is the World University Network that where we address at least two of these issues, the drought and the heat wave, and the impact that they have in food security. We do it as a compendium of researchers throughout the world, and I don't know if you want to pass this around, just to see some of the results of the work done in this network. Today, I'm going to focus on one of the aspects that we investigated within this network, and this is an important and a very relevant process, which is the symbiosis established between the soil-borne bacteria, rhizobia, and legumes roots. Symbiosis, or biological nitrogen fixation in this case, is the second most important biological process after photosynthesis in Earth. And this occurs in special houses named nodules that the legumes roots forms to host the bacteria. The bacteria is there within nodules able to fix nitrogen from atmospheric nitrogen into usable nitrogen by the plant, say ammonia and glutamate. In exchange, the bacteria get some of the carbohydrates that the plants produce as a photosynthesis. So this is a real partnership between a plant and a bacteria. And I want to highlight one of the major contributors on understanding the importance of legumes in soil conditions, in soil health, but also in drought conditions, which is George Washington Carver, which if you want to know more about this black plant scientist, check a biographical kind of review that I did for Current Biology quite recently. But symbiosis is not an easy process to establish. Although it's so important, it does require several steps. And all these steps are regulated by the plant and the bacteria. So the first step is communication, in which the rhizobia basically exudates some chemicals that are then perceived by the plant and leads to the root hairs to core around the bacteria. Then the bacteria create an invagination of the membrane that is named infection thread. And this infection thread gets the bacteria into the deeper tissue. Simultaneously, inside of the plant cell, the cells start to divide and form these, what we know now as nodules, where the bacteria is hosted. And there is where the bacteria can become a bacteroid and fix nitrogen. The plant contributes to this relation by expressing in this region a protein that is named hemoglobin. Hemoglobin, as in humans, basically reduces the amount of oxygen in this region of the nodules, which again plays a key role into nitrogen fixation, as the nitrogenase of the bacteria is really very sensitive to changes in oxygen levels. So this process really involves a lot of communication. Communication between the epidermal cell layers and the internal cell layer where the bacteria is hosted. And this communication is mediated by multiple transcription factors, some of them that are expressed in both of these cell layers, such as nodulin section 1, which is expressed both in the epidermal tissue and in the cortical tissue, where the bacteria is going to be hosted and where the nodule is formed. So we wonder, is this process then dependent on communication via Plasmodesmata? And to do that, we use a model plant that is Medicago truncatula. This is a legume, and they form lateral roots and nodules in response to rhizobia infection. So we look for enzymes that will be involved in callose biosynthesis or degradation that will be expressed in these nodule regions. And to make the long story short, we found a beta-1,3 glucanase, so a callose-degrading enzyme, expressed in this nodule region and also co-expressed with the infection thread here in purple, which drive the bacteria into these internal cell layers. More excitingly, ectopic expression of this beta-1,3 glucanates leads to an increase in the nodule number, whereas silencing using RNA lines of these beta-1,3 glucanates leads to a reduction in nodule numbers. And you can see this phenotype here, where you can see more nodules in this ectopically expressed root of beta-1,3 glucanase in comparison to control. This doesn't necessarily mean that these nodules are functional or that fix more nitrogen. It just means that the plant is able or has the capacity to form more of these houses to host the bacteria. We can also look at callose synthesis. So what happened then when I ectopically express the callose synthase? So, and As expected, when I topically express the callose synthase 3, an activated version of the callose, which again increase callose deposition around the plasmodesmata, you see a reduction in the number of nodules. And the few nodules that are formed in comparison to control are poorly infected and form these strings of bacteria that are basically abnormal. So this suggests that basically this process of nodulation is affected in both cases by topically expressing a beta-1,3 glucanases, by silencing a beta-1,3 glucanases, but also by inducing callose biosynthesis. And this is related with the expression of transcription factors that are in those roots, such as NIN and NFYA1, which are controlling the process of nodulation here in the cortical tissue. And indeed, we could confirm that the expression of the nodulin section 1, a critical transcription factor that was expressed in both in the epidermal and the cortical tissue, is defective when I topically express the callose synthase 3 or the callose-inducing enzyme. So that data bring me to this model in which we hypothesize that rhizobia induce beta-1,3 glucanases to open plasmodesmata, which leads to the movement of transcription factors or of upstream elements of transcription factors to be expressed in both and coordinate both the epidermal development and the nodule development. And now we are starting to look at other processes in which, other symbiotic processes in which this could be important. Here I'm showing you one. Let me see who knows what I'm showing here. So far, it's very similar, isn't it? You have an infection thread. It comes between cell layers. It gets into cells. It moves apoplastically, find a way to get in. And here it comes what give it away. Which is the formation of these feeding structures that basically are all over those cells. Does anyone knows? Yes?
Is it forming arbusculars?
Yeah, they are forming arbusculars. So it's arbuscular mycorrhiza symbiosis, which again have a very important role in the acquisition of nutrients and water from the soil for plants. And this is more generally distributed in all the plant kingdom. So it's not only for legumes. And in exchange, again, the arbusculars want to have the carbon sources from the plant. So we are just starting to look at this arbuscular mycorrhization and how plasmodesmata or symplastic communication can be also involved in regulating this other type of symbiosis. To my final question, which relates to the biomaterials. So how we then can use this information to look at sustainable biomaterials? So we now know that callose is a key player into the regulation of these channels. It accumulates around plasmodesmata and either close the channel or make it more stiffened. In any way, it does modify the transport through these channels. Callose is only expressed 0.3% in the plant cell wall, so it's very, very minor component of the cell wall. But increase in response to bonding or other damage, and also in response to stress. So a way to valorize agricultural waste, which has been extremely stressed, it will be to extract this polysaccharide. Another way to obtain callose is from yeast and from seaweed. So these different organisms also accumulate beta-1,3 glucans, very similar to callose. And in fact, this has been commercialized as immunostimulant And because it contains, this seaweed contains about 35% of their cell walls are made of beta-1,3 glucans, a very similar structure as callose. So one of the projects in the lab actually look at how we can also use callose to or extract this callose from this seaweed or these other structures. But another thing that we are looking is basically to create callose cellulose gels and study what are the properties of this interaction between these two polysaccharides that occur in cell wall. So this now is a very complex slide because the animation has gone. But basically, what we do is obtain callose, in this case from a fungi, and mix it with cellulose in different concentrations, and then study the structure of these mixtures. And you can see here that different callose cellulose concentration have different structures until under electron micrograph. Then we use these structures and tested using nanoindentation, and as explained before, using AFM, we can understand what are the mechanical properties of these gels. And we can see here that when we modify callose concentration, when we increase callose concentration, we reduce the stiffness of these hydrogels, but displace the point of failure, suggesting basically that this callose is making these hydrogels to be more elastic, but also less breakable, so more ductile. So recently we look at what are the reasons why this happened, why callose is able to modify in that way the properties of cellulose. And we use molecular simulation, basically supercomputers in collaboration with a computer scientist, Pietro Ballon. We simulate what are mixtures of cellulose and callose in hydrogels in different concentrations. And we can see here in a snapshot of these simulations, in red here you see the cellulose microfibers, in blue you see some cellulose change, in yellow you see the callose. And you see that they are strongly integrated after 100 nanoseconds of starting the simulation, producing a number of hydrogen bonds between cellulose and callose structures. And these differences in the hydrogen bonding also lead to a reduction in the young modules or in the elastic module. So these basically support our research that elastic properties of callose cellulose hydrogels increase with the callose concentration. So if we look closely to these molecular simulations, we can see that indeed callose act as a glue between these cellulose nanofibers. And here you can see callose in yellow. When we actually kind of float the system, this is all modeling computationally done. When we float the system with water molecules, we see that basically the water molecules are strongly associated with the regions in which callose are. And if we do exactly the same, but with only cellulose, the water molecules will stay outside of this region. This gets us to the hypothesis that callose glue the bundles of cellulose nanofibers and creates spaces for water uptake or water loading. And our hypothesis was that that would lead to hydrogels that have an increased hydration. And that is the case. So if we actually study the hydration properties of this hydrogel by looking at water uptake in these hydrogels and different concentrations of callose, we see that there is an increase in the water uptake by hydrogels that have even 20% concentrations of callose in them. Because water also brings with them dyes, we also look at dye uptake. And what this picture shows is that increasing callose concentration in this hydrogel indeed increase the dye loading capacity of the hydrogels, and also it slowed down the release of the dye. Here in orange is the hydrogels with callose concentration of 20%. So why this is useful is because this becomes a drug delivery opportunity. Cellulose has been used for years as a drug delivery system and in tissue engineering as a scaffold for creating new joints, for example. So by actually adding a very small amount of callose, I get drugs that are delivered or released much more slower. So for certain type of drugs, this could be a system of use. So to conclude my talk, I want you to take home few messages. Has been a very challenging talk, it's again very long, but I want you to think about Plasmodesmata as conduits for intercellular molecular transport. I want you to know that cell walls regulate Plasmodesmata aperture, and by doing that, it regulates signaling in processes that are very important, such as nitrogen-fixing symbiosis. But other signaling processes, such as drought or nutrient uptake, are also affected by molecular transport through Plasmodesmata. I also want you to know that callose and cellulose interact to form elastic biopolymers. And these biomaterials could be used in tissue engineering or as drug delivering system. And recently, I collaborate in this project in which we increase callose concentration in poplar tree. As you see here in the immunolocalization, that this would contain high amounts of callose. And this, in collaboration with Matthew Burdon and Hika Hilariuta in the Sainsbury lab, is a way to basically obtain already callose integrated with cellulose. And if you don't get anything from this talk, I hope that you remember this river of molecular signals between walls, this beautiful view of the Caminito del Rey that I very much encourage you to visit. So I want to thank all my team, which I already mentioned. So my collaborators are here. Paul Knotts, Mike Rees, Simon Connell are mainly leads on cell walls, physics. Pietro Ballon, which is in Dublin, is generating the molecular simulations. Hika Hilariuta in the Sainsbury lab and in Helsinki now is also working with the callose synthase 3 and with the poplar version of it. Fernanda de Carvalho Nivel in Toulouse is working with the symbiosis, and there is a number of funders, and I cannot leave here without telling you about this initiative that me and other Black researchers in plant science have initiated, and that I encourage you to share with your peers if you are not identifying yourself as a Black in plant science. So hopefully we can get a network in which we see more representation of Black faces in this auditorium. So I hope that I don't have to tell you the importance of diversity in plant science. So thank you very much. And with that, I'm hoping that you go away and get some smart questions from me.